Thank you, everyone, and welcome to the last webinar for this year in our Lunch and Learn series. This has been quite fun for us to put together, and it seems that a lot of people have been enjoying it, including being able to watch the recordings back as well. We'll probably run this again next year, and I will drop a link in the chat at the end of this with a little form that you can fill out with suggestions for topics that you'd like to see us explore next year. So just a reminder that this is recording. You are welcome to keep your videos on. You're welcome to interrupt Kent throughout the presentation, ask questions if you'd like, or you're welcome to put them in the chat. But just if you have your video on, be aware of what's going on, in case it shows up on YouTube later. I will, at the end of this talk, probably tomorrow morning, I'll send out the recording to everybody so you can rewatch stuff if you want, or you can pass it on to other people. So today we're excited to jump into how to tell apart our three swallowtail tiger species. And Kent will be diving in and telling us all about how to tell them apart.
Great. Thank
So
you.
Kent, go ahead.
Thanks, Dana. Yeah, I'm going to talk a little bit about tiger swallowtails, but first I'm going to talk a little bit about maybe some of the stuff that people on this webinar have already heard, but remember that a lot of people watch the recording, like a lot of people. So I try to repeat some of this stuff for people that are watching the recording too and might not attend this. And so we're going to jump in about tigers here in Vermont, which I'm calling tiger tails for two reasons. They have tails, but also it seems like the story keeps changing for tiger swallowtails, and I feel like it's sort of the story of science. Science keeps marching on. We keep coming up with new stuff and then either proving it correct or wrong. And I think we're getting closer and closer to understanding for once tiger swallowtails, at least here in Vermont and in this region. But first, I just wanted to go over a little bit about this atlas. This is the second butterfly atlas for Vermont. We did the first one back in 2002 to 2007. And so now we're now 20 years later plus doing the second butterfly atlas. And I always go out on a limb and say this. I think we're the only state to do two butterfly atlases so far, which is kind of cool because, well, I think it's really cool actually because we actually are going to be rewarded from all that work we did in the first butterfly atlas by being able to see changes after 20 years or potential changes. Both may be bad and both may be good for species and their ranges across Vermont. So we actually will have population level changes and range changes that we can look at after 20 years. So we dreamed about that in the first atlas, and here we are doing it. And so, how do we atlas? All of our major atlases follow the same kind of similar protocol from the first breeding bird atlas up to all those atlases you see on the slide to the third breeding bird atlas, which is going to start next year. Um, and they use this statewide atlas sample scheme, which has these priority blocks. And there's 184 of them you can see on that map, randomly selected across the state that are the minimum number of survey blocks that we need to complete during an atlas to say the atlas is finished, that we've done enough to survey enough of Vermont that we have a sample. And so you'll hear us all the time, maybe ad nauseum, that we need to get these 184 priority blocks done. And so my house is not in a priority block, but I've done really well. I think I have 41 species in the block that my house is in. But I have priority blocks all around my house that I've adopted because really to get this atlas done, we have to survey all 184 priority blocks enough to feel like we got a good sample across the whole state in a 5 or 6 year period. So we're really trying to get folks to really consider working on these 184 priority blocks as much as you can. And that can be either by adopting one of these blocks, which is like what you do if you're really going to get a lot done on the butterfly atlas. And so you can go into our block mapper tool, click on a block, sign up for the block, you've adopted it, and then you're committed to like trying to survey it a lot in order to try to attain at least 40 species on that block. And for some blocks, that's really hard to do, but that's sort of our upper bar. Like if you hit the 40 species mark on a block, we feel like that block has probably been surveyed well enough. But we also know that not everybody can adopt a block and wants to go, butterfly wild, but really wants to still contribute. So I really like to tell everyone like if whether you're watching butterflies in your garden or you're way up there adopting several blocks, it doesn't matter to us. We want you to be part of the Butterfly Atlas because every drop of data helps us understand put together this sort of puzzle of butterfly population change in Vermont. So whether you're adopting a block and checklisting and using eButterfly, where you're timing yourself, recording how far you've walked and counting every butterfly you've seen and putting it on a checklist, or whether you're just using presence only and maybe putting it in iNaturalist, like, hey, I saw this butterfly on my walk with the dog or something. It's all good. We want all that kind of data for butterflies. So I really encourage everyone, whether you're just collecting butterfly data here and there using iNaturalist while you're doing something else, that's awesome. I'm totally into it. I hope you share your data and help the Atlas. Or whether you've adopted, 3 blocks and you're just every weekend and every waking hour thinking about butterflies, that's amazing too. So I really try to tell everyone there's something for everyone in these atlases. You don't have to be an Uber butterfly watcher. It's amazing if you are, that you don't have to be. Everyone can contribute. And so these blocks, as you can see, they're like 3 miles by 3 miles. There's plenty of space, maybe sometimes too much space. You can ask your neighbors if you can go in their fields. It's fun to explore these blocks and find really interesting things. So that's kind of how the blocks work. And you'll hear us all the time, if you haven't already, like, hey, have you been to a priority block? And the last thing I'll say about the priority blocks is you don't have to adopt a priority block. You could just go to one and butterfly on it. So maybe a weekend comes up and you're like, you know what? I feel like driving up to Franklin County and going to a block and butterflying in a wildlife management area or along a class 4 road. You should do it. Go for it. Go for a little road trip and see if you can find a bunch of butterflies on another priority block that you haven't even adopted. That's helping us get towards that goal of surveying these blocks enough to feel like we have a really good sample. So that's my spiel about how atlases work really top level and how important it is for us to focus on these 184 priority blocks to make sure we get a good statistical sample across the state. The other thing I'll say real quick before I jump into taggers is that maybe not real quick, I might spend a little bit more time on this, which is really improved some of the second butterfly atlas tools recently. I just wrote a priority block checklist tool and we sped up the block mapper tool a little bit. And I just rewrote the flight times tool to be faster. And so all these now are links at the Vermont Butterfly Atlas website. Right on the menu, you'll see them, all the tools like they are. And I really wanted to highlight first the priority block checklist tool. And I'm actually going to go to it right now live. Um, and so if you go to it, here's all the tools, the priority block checklist tool, the block mapper tool, and the flight times tool. And this is the priority block checklist tool. It actually, you can get a checklist of what butterflies are known for during this atlas from any block, but it prioritizes, again, the priority blocks. And you can see over here, like the different color codes for how many species we have on them. And then the non-priority blocks. And you can turn these off. So if I turn off like the blocks that have just less than 20 species, I just do this to illustrate like there's a lot of blocks right still after 3 years that have less than 20 species all over the state. And some have 0 species recorded on them. And so when you see that, those are like primed to either adopt and help the person that's already adopted it, more than one person can adopt it, or take a road trip and go there one day and help get blocks. But the cool thing about it is you can zoom in to a block and say, let's say this block right here, Woodstock South 1, which is my block. It has 34 species on it. If you click on the block, it's kind of neat. Um, if it's going to work, I'm on the internet. There we go. It's kind of neat. If you click on a block, it'll tell you, here's the block. It's got 34 taxa recorded, and you can download a checklist PDF, which is kind of neat. But you can also just scroll through and see. So here's 33 species recorded, and here's everything I found so far. Plus, if you go scroll to the whole way to the bottom, you can see here's a genus only record. It wasn't ID'd to species yet. So that's why it says 34 taxa. And then most importantly, this is not live data. I have to update this app every week or two, which I'm going to try to do. And so the data is up to the 14th of May. And so I'll keep updating it and that number will keep changing. So you can see like when it was last updated. But the neat thing is I think is you can download a checklist. And if you download it, um, oh, I didn't set up to see it. I just downloaded it and it's a PDF and it shows every butterfly in order in the state of Vermont and it automatically checks off the 34 taxa already recorded. So you can download that, print it out on your printer. It's just black and white. And then as you work on that block, you can check off with a pen the new species you're finding and keep track yourself also. Take it out in the field with you, maybe pre-study some of the species you're missing and remember like, oh gosh, I'm missing black swallowtail. I can get that. I know that's going to be in so and so's garden, or I can find it in this field. So you can like pre-plan by having downloading that checklist on here. And you can do it for non-priority blocks too. So you can click on any priority block or mouse over. You can see that South Royalton 6 has 5 species on it. You can click on those too. And you can play around, you can zoom in. And then also you can switch from the base map. You can switch from just a regular map to a satellite view. So you can zoom in and see things on your block like, oh, there's a field, there's a beaver meadow if you want to, and that kind of thing too. So this tool really is about like getting a checklist and keeping track of how many species you're finding on a block you might have adopted or a block you're going to visit. And so let's say I'm going to go visit this block tomorrow. I can go 31 species. What are they missing? I can target species to try to see where I need to go. So it's a fun tool to use. The block mapper tool is the same as always. It's live data, so it loads slower. But this is where you can go and see and sign up for a block. You can click on a block and sign up for it. It's just a Google form. And then you can also click on a block. For example, I'm just going to zoom in and click on any old block. You can get a species checklist here too, but it's live data and it's a little slower and it's not set up as nice. But you can compare the first atlas here to the second atlas, which is kind of cool. So you can be like, oh, in the first atlas, they found 42 species, and here's the ones I'm missing. So this is a neat tool to compare the two atlases. And then the last tool that this one we just updated is a
And
flight test.
just
Yeah.
before you continue on to the flight times, we had a question about how the priority blocks were originally chosen.
So the priority blocks were chosen by using back in 1977 for the first breeding bird atlas. They took the 1 to 24,000 quadrangles for topographic maps for the Vermont, and they divided them into six, so 6 blocks in each one, and that's like a giant grid across the entire nation. And then out of those, the 6 blocks, they randomly chose a random number 1 through 6, and that was the priority block for that quad. And so there's 184 quadrangles, and so there's 184 smaller priority blocks in those quadrangles. And now, fast forward to modern times, that's what every single breeding bird atlas does across the country and butterfly atlas. And next year, when we start our breeding bird atlas and we're using eBird breeding bird atlas, you'll see that system is used by every single state, so it's all comparable and all using the same grid across the whole entire country. So they were randomly chosen quad by quad. And that's how they were chosen. Back in 1976, I think actually is when they did it. Um, so the last tool real quick is the Vermont butterfly flight chart. It will automatically can do all kinds of stuff up here, change to a heat map, change to a chart. I just rewrote this whole thing. You can get different metrics, individuals. You can sort it differently by you want. You can click on, it'll highlight this week, and you can click on the highlight and it'll sort it by the most abundant to the least abundant. So you can see what's flying right now across, it's all of Vermont. So you can get an idea like, oh, I'm probably not, I have a smaller chance of finding a question mark underneath my question mark than I would maybe a green comma right now, maybe. But just as an example of this is just how many individuals we're seeing over time on those each week by week. So it's a great thing to be able just to quickly get a flight chart and be like, yeah, I'm probably not going to find, I have a good chance of finding a red admiral, maybe an American lady, but maybe not a painted lady. So the flight chart's just a great way to study and check to see what's flying now and what might be coming up. And you can see what different species have 2 flights. So American copper has a couple of flights. You can just quickly scan down and see like, oh, here's a double flight. What is this? Common ringlet. We all know it has an earlier flight, then it's a period where there's not venting, then it has a second flight. So you can see the different flights here. So it's a fun little tool. So those are our updated tools. Um, they're really fun to get lost in rather than watching Netflix or something at night. You can go and look at butterfly tools and have fun with those. And again, I'll try to keep the data up for the block checklist tool updated at least bi-weekly, if not every week. So those are some new tools. All right, on to taggers. Unless anyone has questions about tools, you're free to ask them now or at the end of this, or anytime. Email anytime and ask questions or whatever. So we'll talk about taggers. So during the first atlas, back, like I said, over 20 years ago, we had three tigers, and we had Canadian tiger swallowtail, which had a spring flight right here. And then we had this thing that happened after Canadian tiger swallowtails disappeared. We had this flight of tiger swallowtails in summertime, and it had we just was like, it's a weird beast. We don't really know what it is. And we just called them tiger swallowtails. We're like, just a weird tiger swallowtail that nobody knows what it is. And we suspected it could be a new species. It could be a hybrid. There was a lot of like speculation what it was. And I remember, I bet Kevin Hemian isn't on this call, but I remember one time being out with Kevin during the first Atlas, and he was saying like, you know what? I think this summertime tiger swallowtail, it behaves differently. It flies different. It has a different behavior. And like, I didn't notice it, but then I started noticing like he's really observant, and I'm like, wow, he's on to something here. And so we had this weird tiger beast. And then we also had rarely eastern tiger swallowtails, which were from further south. They didn't breed in Vermont, and we didn't really find them that often. And so this is what we had in the first atlas. So we had Canadian tiger swallowtails, some weird thing that flew after that, and then scattered about were these eastern tiger swallowtails that were from further south and would pop up in Vermont. And we just kind of dealt with it. We just left it go of like, yep, someday someone's probably going to describe that tiger swallowtail. And it had tons of names. People called it, oh, it's just Eastern tiger swallowtail. Oh, it's a false second generation of Canadian tiger swallowtails. It's just a late flight. It's a hybrid. It's like you can see all these names I've found. It's an app. Someone once said it's Appalachian tiger swallowtail like, almost like they wanted it to be an Appalachian tiger swallowtail. And then finally, like the Canadians and Ontario, I started hearing them call it the midsummer tiger swallowtail. And Chris Schmidt up there, who is an amazing lepidopterist and taxonomist, was really starting to study them hard and was putting out little papers and reports with the Toronto Entomological Society that were really insightful, saying like, yep, this thing is a different species, and I call it the midsummer tiger swallowtail. And then lo and behold, that's a picture right there you're seeing of the midsummer tiger swallowtail, the type specimen picture right there. Last year, a paper came out, the cryptic new species of tiger swallowtail from Eastern North America. You can see Chris Schmidt's name at the end there. They teamed up and put this paper out, which was widely expected, and they presented molecular data, DNA, like different phenologies of all the different species, all these morphology things and natural history, all in this paper, which thankfully is open access. So if you're really into like going to the source after this and reading this paper, you can just download the PDF and read. And even if you're not into reading science, peer-reviewed hardcore science papers like this, it's kind of fun just to read through it and see what goes into describing a new species. I've read this paper. My copy of this in my hand here, dog-eared, destroyed, and tattered because I've read over this paper so many times, and I still every time I read it, I'm like, oh, I must have missed that little piece. It's filled with stuff in here, and some stuff that, frankly, I don't really understand. Some of the DNA stuff, but it's really interesting to read. And so I've done that to try to figure out like how can I learn how to deal with these tiger swallowtails in Vermont now, and go back and redetermine a bunch of the tiger swallowtails from previously. And so it's a work in progress for me still, and I know it will be for a lot of you. And so, like, I try to be humble about when I'm IDing these things on iNaturalist and eButterfly. And so I will say, like, they're not easy. And it's going to take a lot of different, if I scroll back here, it takes all these things. Like you have to look at phenology, you have to look at some of the morphology, and maybe some of the natural history too. Obviously, we can't deal with molecular right now because we're just watching butterflies, but in the future, hopefully there'll be a marker molecularly too. There isn't exactly one yet, not a perfect one. So I just try to remain humble. If you're IDing your tigers, like I'm gonna say right off the bat, like if you feel overwhelmed and you're not sure, like, oh man, it's like late June. This could either be a Canadian tiger swallowtail or a midsummer tiger swallowtail. Like both eButterfly and iNaturalist have a complex on there. So you can put in like Eastern Tiger Swallowtail complex, which means like, I don't know, it's one of the tiger swallowtails, and I can't either tell from this photo because the photo doesn't show me the right things, or I don't actually know. It's okay to put down it's one of the tiger swallowtails. It's in the complex. Because someone like me will come through and see if we can ID it, and there's a bunch of people like me and we might not be able to either. And so, and there's a ton of them on there where I just put complex on because I'm like, I'm not really sure, or this photo isn't good enough for me to tell. And so I'd rather have us put in like it's part of the complex rather than putting a species down if we're not pretty darn sure what we're looking at. So that's my spiel about IDing these things. It's not, it can sometimes not be easy, and sometimes you have to walk away from them for a little bit. But we have three species now, still. But now we have a name for the middle one, and it's officially described and it's pretty widely accepted for at least for here in Vermont. So now we have Canadian tiger swallowtail. We've got midsummer tiger swallowtail, with its official name, and Eastern tiger swallowtail, which is still honestly quite rare in Vermont, not found that often, and probably doesn't breed in most of Vermont or doesn't survive the winter in most of Vermont, probably as much as a little bit further south in here. But that said, like down where Terry is and Kevin down in the the banana belt of Vermont, like they could be there as breeders too. So they're really close by in Massachusetts and things are warming up. So I don't want to say they don't breed in Vermont, but they're really uncommon. But again, some of the key features here morphologically are not are the first key feature is going to be phenology, and I'm going to get to into that in a second. But one of the key features here what all these lines are pointing to, and you're not going to learn them on this slideshow real fast, but you can come back to it, and I'm happy to share this with people. But this is all taken out of Chris Schmidt's information, and I've bugged Chris a couple times from that paper I showed you. It's all from that, and then from just looking at a lot of them and trying to start to figure it out. And some of it, it's a little bit of like after you look at a bunch of them, you get it. Um, but the first thing to look at is this upper area of the wing here. And it's almost always sort of a continuous yellow for both of these two species. And if you look over here at Eastern, these are the cells that are actually separated by black a lot. And so, and you can't just take one thing. You got to take it all. So we're going to talk about phenology. That's an important thing next. But this is just the key features. So you'll notice though, on every one of these pictures, I'm showing you two things. One, I'm showing you these are all males, and I'm showing you the underside. And so you don't necessarily have a male, but there's extra features on males that are helpful, I think. Females are ideal too. But the key here is that I'm showing you the undersides. So if you take a picture of a tiger swallowtail that's at the tail ends of its flight period, and you only have the top of the tiger with its wings spread open as your picture on eButterfly or iNaturalist, then anybody else is coming in to help determine that species, to help identify it, they're not really going to be able to. You really need to see this shot right here, the underside. So you either have to wait until the butterfly closes its wings and is nectaring like this, or you need to catch it and take a picture of its underside real quick and then let it go. But this is the shot you really need to have for some key identification features. And so it's this piece right here. It's how straight this line is right here. So you can see it's really straight in a Canadian tiger swallowtail, slightly maybe not straight in midsummer tiger swallowtail, and there's a bend to them. They're like a little concave. Um, if you look at these little lentices right here, these little marks right here. They're kind of, now bear with me, but they're kind of squarish here, or rectangular, I should say. They're rectangular here. Sure, they got a little curve to them, but you can sort of see a rectangle. And here it's sort of a rectangle. Yes, they have these little tails going off of them, but they're kind of rectangle here. Here, they're definitely going to be like crescent or rounded off. They're different. Um, it's subtle, but start looking at a bunch of them. And we're going to show you how subtle it is in a second. But then probably the biggest thing is this band right here, this anal band right here on the underside, on this lower cell. And you can see how thick it is here on Canadian tiger swallowtail. And it's a little bit thinner on midsummer tiger swallowtail. It's up to around 50% sometimes, down to maybe 30%. Here it's like more than 50% usually. It's like really thick. So think colder times of the year, more black. Warmer times of the year, less black. And then you get down to really warm places, eastern tiger swallowtail, this thing is usually like really thin. Um, and so those are the key features and why you have to take a picture on the underside of it. The problem is like I'm showing you pictures here that are kind of definitive. And if you start to look at them in a second, we'll see some, it's really hard to
Kent,
see.
a question before you move on.
Yeah.
What are some of the ways that you can tell the difference between the males and the females?
Oh gosh, that's a really good question. So I used to think like the top side, the females have more blue wash on them, but it turns out like with Canadian and midsummer tiger swallowtails, that blue wash kind of isn't there as much. And so you can see like the females have more color. But really what I like to do with butterflies this big and even small butterflies is like you can really look for claspers. And I have a picture later that shows the close up, but I'm pointing right here to this clasper because I forgot to mention that. And I love whoever just asked this question. If you see these claspers, and I'll show you the difference between a male and a female clasp without claspers, but males have claspers to hold on when they're mating. And it's like this little, honestly, it looks like a little fin on the end of here. And females just sort of end and they don't have this clasper. Well, it's very helpful for IDing too, if you get a close-up of that, but not between midsummer and eastern. Their claspers are both the same. But between these two and Canadian, you can tell the difference. The claspers on Canadian tiger swallowtail, which I'll show you in a little bit, have dusted black scaling all over them. They're not all yellow. But midsummer and eastern, they are bright, bright yellow with hardly any dusting of black in them. And so you could literally probably take a picture of a tiger swallowtail here and just give me the clasper, and I can tell you whether it's a Canadian tiger swallowtail or not, just by the clasper. So it's the claspers, and I'll show you male versus female in a slide in a second. And I skipped over that little detail. Taking a picture of the claspers is a great idea then, if you have a male. So we got phonology, we're going to have morphology, and we also have to think about range. And so this is actually the range map out of their paper that they presented. And so we have this hot pink, this pink right here is where they feel is what they have described as the main range for the midsummer tiger swallowtail. And so I pulled Vermont out and put it over here, blew it up a little bit. So you can see here's the range that they expected midsummer tiger swallow to be in Vermont based on all the samples that they looked at. Doesn't mean this is right. It means this is what they think it is to the best of their scientific knowledge at the time they wrote this paper. But they also put on it where is canadensis, where is Canadian tiger swallowtail, and where is glaucus, where is eastern tiger swallowtail. So horizontal lines, horizontal, we can see this is the whole range. So all of Vermont has canadensis in it, going down in through Pennsylvania in the mountains, down on the high level. And then the diagonal lines are glaucus eastern. And so you can see over here in this map, you can see the diagonal lines. Well, okay, eastern tiger swallowtail is right over in Massachusetts. And if we look at the blow-up map, I'm pretty sure eastern tiger swallowtails don't know there's a border right there, but their diagonal ends at the border of Vermont. And so we need to learn a lot more about what's going on here. This is like a zone where all these species are coming together. Likewise, we need to learn about what's going on up here too. This is a zone where only one species is putatively and none of the others. So the paper, this is the best science they put forward on this. But when we start to look at it critically, we realize like there's a lot to learn even here in Vermont. Not to mention that, in Ontario, they don't have the elevation we have. So what's going on with elevation with these species? Is Canadian tiger swallowtails hanging on later at elevation, phenologically, and maybe midsummers don't get up to certain elevations? We don't know. So yes, we can take into account range in this too, but we also have to understand that we need to learn a lot about range too. And that's beautiful part about this atlas also. We're going to learn a lot more about what's going on with these tigers, both in what we've done in the past and what we're doing right now. If I look at this really quickly, just for this talk, this is data I took from 2010 that I ID'd enough stuff to put it in here. So red is Midsummer Tiger Swallowtail, and blue is Canada Tiger Swallowtail from 2010 to now. And then I just brought out all these different all these biophysical regions, these different regions of Vermont based on biology and physical zones. And you could kind of see that, like, hey, they were right. There's really nothing in the Highlands. The Northeast Kingdom there's one record I found close to it. That's it. So far, we have zero records for the Northeast Highlands, the Northeast Kingdom Highlands, um, for Midsummer Tiger Swallowtail. So that would be something like in July, like, hey, can we start picking up tiger swallowtails in July? And maybe we're only picking them up along the Connecticut River. Maybe they don't like to be up higher. We don't know. Black box there. So this is why it's important for us to understand what's going on with the tigers. And there may even be something going on here with elevation. We can see in the northern greens where there's no midsummer tiger swallowtails. Is it because we haven't found them yet, or because we haven't looked yet? We don't know yet. So that's why these priority blocks come in to play, so we get a good sample and start to understand this. So looking at this, it's following generally what they said so far. Um, and then I just quickly looked at, just for the heck of it, because I mentioned elevation, I took that same data and I'm like, is there an elevation signal yet, just real quickly in our data? And so you can see, like, we have over 1,000 Canada tiger swallowtail records, 340 some Midsummer Tiger Swallowtail records, and you can see, like, not really. I mean, maybe Canadensis is a little bit higher elevation, but these box plots right here are basically showing that they're pretty much both using the same elevation. So far in the sample, looking um, quickly at these samples, like, there's no elevational gradient going on that I can see. Doesn't mean we won't find it, and we need to do more of this stuff, but it doesn't look like it's happening so far in our sample. So back to morphology. They examined 22 different traits, and you can go to the paper and look and decipher every single trait that they looked at for Midsummer tiger swallowtail, Canadensis, glaucus, and then I cut it off, but also glaucus is the eastern tiger swallowtail. I cut it off, but also Appalachian tiger swallowtail is on here, and another tiger swallowtail, which is not part of Vermont. And I've gone through this thing a bunch of times, and some of these things are not very useful, and some of them are really useful. And so what I've tried to do then is take all that useful stuff and bring it into, like, what traits can we really look at? And so I already showed you a picture of some of like the actual physical traits on the adult butterflies that are useful, things that are in here in their language, like hindwing anal cell black band width on males. Okay, it works on females too, but females are a little bit different. It's more obvious on males. So they measured all the males. So for Midsummer Tiger Swallowtail, that little black band I told you about, it's between 40 and 50% of the cell is black. For Canadensis, it's 55 to 90%. It's a really thick black line. And then for Glaucus, it depends if it's summer or spring. We don't really get very many spring records here at all, if any. We're talking about the summer brood for them, so it's 10 to 40%. So it's thin. That's a really good field mark. Some of these other things, like hindwing tails, I don't find any of that any useful. So out of the 22 traits, I'm trying to point out things that are useful. And one of the traits is phenology. So I talked real quickly about morphology, then there's phenology. And I asked Chris Schmidt recently, I'm like, what's the very first thing you think of when you're looking at a tiger swallowtail? Let's say you're in Ontario or Vermont, and he's like, phenology. He's like, you got to use phenology. It's going to help you, like, to understand it. And so I whipped together the phenology so far for that same data set for us for 3 species from 2010 to presently. And so you can see blue is is the Canadian Tiger Swallowtail. What's flying right now? Canadian Tiger Swallowtail. It's pretty much the only thing flying in Vermont right now. Should you ID it only on phonology? Absolutely not. Would you probably be right? You're probably going to be right if you did. But it's just one of those things. Again, it's a piece of the puzzle for you to ID the species. So it's a big piece is going to be like, it's May. I'm going to see only Canada tiger swallowtails. It's not until we start to get well into June, and some of these might not be ID'd right, some of these tails yet. I'm still going through it all, that we start to get midsummer tiger swallowtails potentially. And so this zone right here, this is where I get nervous when I'm IDing things. So I'm taking phenology into account, and then I skip to morphology, and I'm like, wow, it could really be any one of these things phenologically. So I need to weight the phenology less and really start to look at them. And Chris Schmidt told me something very recently. He said, look, if it's July, the beginning of July, and you find a tiger swallowtail and it's really fresh, he's like, you have to look and realize, like, if it's really fresh, it's probably going to be midsummer tiger swallowtail because it just eclosed. It just came out of its chrysalis. And by fresh, I mean like it doesn't have any bite marks out of it. The scales aren't missing. It looks like fresh as the day it just came out of its chrysalis, like maybe it just did. Versus at that point, by the 1st of July or even the last week of June, most of the Canadian tiger swallowtails, they're going to look beat up. They're going to look like they've been through a little bit. They're going to be missing scales. They're going to look roughed up, rough around the edges. A tail might be bit off by a bird. Like they're not going to look good. And so he's like he keys in on the condition not only during this period too. Like what is that thing looking? And then starts to look at morphology. But you'll also look at this Eastern Tiger Swallowtail. Only 25 records. Again, assuming they're ID'd correctly. I still have to go through some of these. And you can see there's a couple early here, putative records. But it isn't until like near August that we start seeing a lot of, not a lot, but a few Eastern Tiger Swallowtails rolling in. They're probably coming from south. They're coming northward and rolling in. So that's when we have to start looking for the unexpected. Like, hey, especially if then you start thinking about now you're thinking about phonology and the range map. Where am I? Oh, I'm in southern Vermont. Whoa, I'm pretty close, if not where they're eastern and also midsummer tiger swallow. I have to take into consideration now eastern and midsummer. I'm probably not dealing with Canadian tiger swallowtail. So this is telling us a lot here. Phonology and where we are and the condition of the butterfly before we even get to some of those key field marks through morphology. So I showed you the three tiger swallowtails, and I'm showing you again, just viewing in. So this is that underside of the forewing. So here's midsummer. Here is sorry, I keep skipping from Latin to common name. I'm sorry. This is Canadian tiger swallowtail, and this is Eastern tiger swallowtail. And these are just different butterfly individuals. So you can see this is right out of their paper. You can see like there's some difference. Like look at this one. It looks a little bit like this one. So there is a range of glaucus. You can't just use this one mark. But if this mark is like this, you're not going to really see that for one of the ones above. So the extremes are really important. The overall trend is important. So you can't just use one field mark, but that one field mark could be really helpful. The same thing if you look at this again, that band I talked about, this anal band right here. Here is the same thing in the same order. You can see how these are really thin. Doesn't mean one doesn't have one that looks a little thicker. And this one's a little thicker, and this one's a little thinner. But overall, you're going to find out that canadensis is the thickest, midsummer is medium, and eastern is thin. Again, not using one field mark, we're putting everything together. And so there is a variety of life here. And then here's the claspers. And I only show you two because midsummer tiger swallowtail clasper on males looks the same as eastern tiger swallowtail, unfortunately. And it looks really yellow like this. Versus Canada tiger swallowtail has got a lot of black scaling mixed in. And you can readily see that with your naked eye. If you catch him or you take a picture and zoom in on it, you can see this. It's really quite obvious. how much darker it is. So think cold, dark, warmer weather, yellow is how I just kind of remember this kind of stuff. So I really do key in. The first thing I key in on is this anal band here. And so here is a key thing. Um, how am I doing on time? Holy smokes, 15 minutes. Sorry, people. Hope you're still enjoying this. I'll hurry up, Dana. Um, but here it is. Here's the three species. So we have Canadian tiger swallowtail. In the middle, we have midsummer tiger swallowtail. And here we have the summer form of eastern tiger swallowtail. And I think these are both from Vermont, and obviously this isn't. I took these pictures, I think, in Maryland in the summertime. So they're in Maryland. And these are all males. See the claspers on each one of them. Um, and so if you look at this, though, what I'm really pointing out here is the amount of black here. And so it's 55 to 90%. The distance right here is 55 on general, approximately 55 to 90% of the cell is black. And that's canadensis. That's Canadian tiger swallowtail. 40 to 50%. Okay, remember 50 and 55%. Boy, I don't know, unless I'm measuring it, that's pretty close. So again, you can't just use one field mark. But if it's the extreme, then you can. So it's 50% or 40%, which this is here. Then you've got midsummer tiger swallowtail. And then the really thin one, which is 10 to 40%, which is comes up to that, it's just thinner, is eastern tiger swallowtail. But here's the cool thing. This one, if you look at it, and this took me a while, I looked at a lot of these, and I feel like you start to see it better too. If you look at them really fast, turn your head and look at it really fast, is that canadensis, if you look at it really fast, this right here looks like in the shape of a V. It really comes to a point here because it's so thick for some reason it looks, and now that I've looked at so many of them, it stands out to me like a sore thumb that it looks like a V. And both of these are U-shaped. They're thin and they allow it to be more of a roly-poly U-shaped thing going on here rather than V. And I feel like now I've looked at so many of these that I feel like I could do this driving by on my bicycle. If one of these butterflies is nectaring at a plant, I feel like I look over, that's a V, that's a U. I feel like it's even better that way if you look at it quickly. Like I said, turn your head and look at it and don't stare at it too long. But after you look at a bunch of these, you will start to get the feel for V versus U on this, which is also really helpful because then it helps you decipher not just the thickness of the line, but also like, oh man, this is really V-shaped. Um, and again, this is just one of the field marks of a bunch of them that you got to put together to get the whole idea behind what you think it's going to be. Side note, real quick, because I love natural history. Um, here's the thing is that this canadensis, well, both males and females always are yellow. Um, for midsummer tiger swallowtail, males and females are always yellow. For eastern tiger swallowtail, males are always yellow, but females for eastern tiger swallowtail have a black form. And they have a black form because of one thing. They like to mimic. It's called Batesian mimicry, named after Bates, a famous lepidopterist who studied butterflies in the Amazon and elsewhere. And that is that this is actually right here an eastern tiger swallowtail. And if you look through the black, you can see the tiger swallowtail markings behind it. Here's that shape right here. You can start to see, oh, it is a tiger swallowtail, but it's a black female. And it's mimicking this right here, the pipevine swallowtail, which we do have a few records for in Vermont, but they're just accidental records here. They breed much further south of here on pipevine plants, which are poisonous. And so their eggs have poison in them. Their larvae have poison in them from this plant, and then the adults retain the poison. And so pipevine swallowtails are quite poisonous to birds and other things that would eat them. And so it's an advantage to look like this poisonous one if you're not poisonous. And so in this range where the eastern tiger swallowtail is, say down in Maryland or even further south, there will be black form females that are mimicking this swallowtail. And it's called Batesian mimicry because these are not poisonous. They just try to look like this one to try to cheat off of the poisonous model. So this is the poisonous model, and this is the cheater trying to cheat off of it. And so it gets selected for. And then, does anybody recognize these species? Think about them. They are in Vermont, but less so because this thing is not in Vermont. These two species can be found in Vermont. Um, and if you know what they are, you can tell me later, but this is a key field mark for this one right here. And this one you should recognize. These two also are mimics of pipevine swallowtails. So all three of these butterflies are mimicking pipevine swallowtails. Pretty cool Batesian mimicry. The other kind of mimicry, if you're thinking about it, Mullerian mimicry, is when everyone's poisonous and it behooves you to look all alike because then the predators could probably learn that, like, oh, I'm not going to mess with you, you're all are poisonous. It's a different kind of mimicry. There's a little natural history. Um, so let's end this up real quick with a quick test. I'm not going to have anybody shout it out, but you can just like write down what you think or think about what this is. Here's your first one. What do you, I'm not, and this is like I told you at the beginning, like take into account the range and the phonology and everything. I'm just throwing them up here just morphologically to see if you can guess what these are without giving you any other information. Just see if you think what these are. And this first one's a cheater because it has some points to it. And so here's the first one. Just take the next 10 seconds and like look at it and be like, I think it's one of these three species. And yeah, and then I'll shout it out. I'll give you a few more seconds. And I can tell you that this picture was taken in, I want to say that it might have been taken in Virginia. So now you know what it is. I'm hitting at it. So this is an eastern tiger swallowtail. And if you look carefully at it, it doesn't have claspers. It's a female. It's chunky, no claspers. Eastern tiger swallowtail. And so I looked at these are curvy. These are have black going through them. They're not a solid yellow line. These are like circly, not squarey. Oh. And this is pretty darn thin. And if you turn this up on end, it's pretty U-y. Here's another one. I'll give you 10 seconds to look at this one. Again, we're going to the field marks. Let's look at this yellow mark. Let's look at the shape of these. Let's look at how straight or not straight this is. Let's look at our favorite thing here. And like what if you turn this up on end, like you turn your head sideways, what shape is this? And this is a male. It's got claspers, so we can look at the claspers and how thick this line is too. That's another mark. So you just look at all that stuff and put it together. And if you're a really good naturalist, you're like, oh, it's on Joe Pyeweed, and Joe Pyeweed doesn't actually start flowering until later. And so you would recognize that I took this picture in my pollinator garden at my house on about July 25th, and it's a midsummer tiger swallowtail. Um, and it has a U shape. Here's another one. Just quickly look at it. And this one's on milkweed. Um, so that's the milkweed's flowering. So when is this? It could be, that could be in June. We don't really know. This is pretty thick. Um, so you start to piece this one all together. Like, I don't know, it's 50% maybe. Could be a little bit more. It's kind of thick. Um, and then you start to look and like, are those claspers right there? Oh, those look pretty yellow. Oh, these look a little bent. These look a little square. And if you knew the date from this, you'd know it was almost July, and it's a midsummer tiger swallowtail. Um, and then here's another one. Give you a second just to look at it, see what you think. This is like Jeopardy, Butterfly Jeopardy. All right, so you looked at this. I'm sure you did. You're like, oh, this one's turned up so I can tell what letter this is. And everyone said, you know what? I think this is the letter U. And this is kind of thick. And this is a little bit bent. These are a little square and like, and button bushes is going. And, but how thick is this? It's not as thick as anywhere 40% or anything. And it's a little square, but not really square. And these are not as yellow. This is not a yellow line. These are divided and the button bush is flowering. And Kent took this in Maryland, and this is an eastern tiger swallowtail. And then I'll just end real quick here with this tiger swallowtail blitz by saying that I took this picture on Monday. Dana's with me. This is a female, and she probably just eclosed, like literally just came out of her chrysalis, I'm guessing. Super fresh. I mean, look at all this overscaling. This thing is immaculate. And she was really fat and she was still exuding moisture, I think, from eclosing and just learning how to fly around. And I easily grabbed her real quick to get this picture. No claspers. But look how thick this is. And it still looks pretty, it's a V shaped. And look at this, it's all yellow. I mean, this is canadensis. This is a female Canada tiger swallowtail that's about to go lay a whole lot of eggs. And it's very fresh. It probably just came out of its chrysalis on Monday. And then right nearby it, cheek by jowl, I captured this male who was also incredibly crisp, probably just came out. And you can see these claspers here. And if you zoom in, did I do it? Zoom in on the claspers, and you can see all this black overscaling on the claspers. It's not really bright yellow, all this black overscaling on it. And you can see how thick this is, and it's V-shaped, and it's only May. It has to be canadensis. Um, so, what are we going to work on right now in the future? We're revisiting all the records. We're trying to redetermine everything. iNaturalist, eButterfly, museum specimens, all the records from the first atlas are being redone. And so I and others have been working hard at doing this, but there's a lot to do to get everything back up to snuff for today's story about what we think our species are for tiger swallowtails. What you can do is help annotate Vermont records for flower visitations. And if you find any larva feeding, please put the host plant down, and you can do that in iNaturalist. Um, and we're starting to do, like I said, we're starting to compare starting to get into the nitty gritty beyond what the paper did to describe the species, start to understand what's going on with this species and how they're doing in Vermont. And we don't really know even what this thing really feeds on here in Vermont. We know if they like white ash and that's one of their favorite species and it's really good for them to feed on, that might not be really great because white ash is in trouble from disease, introduced species, killing it. Um, but if they can deal okay using quaking aspen and black cherry, great. But we actually don't know in Vermont what they're using. So if you find caterpillars feeding on hosts, we really want you to write that down, but the problem is that we won't know what they are because you cannot ID the larva. You'd have to take it and raise it, and that's a whole different subject, which I'm not going to get into. Um, and finally, your to-do list. I'll skip real quickly ahead. Your to-do list is if you haven't joined the Alice, like I said, join it at any level, whether you're adopting a block or just watching butterflies in your yard. It's all awesome. You're going to love watching butterflies and help report them. If you can, adopt a priority block or at the very least, find a priority block and just go visit it a few times, even if it's a road trip. And above all, if even if you don't do anything, like no matter what you're doing, like watch butterflies, like watch them wherever you are. They're amazing. They're beautiful. They're in more places than we realize when we start watching them. And they're just super fun to watch their behavior and not just go out and just record what they are, but what they're doing too. And that's my spiel. I'm happy to stay on as long as you want for questions. I'm supposed to only take 45 minutes and then questions, but I blew right through that and I'm sorry, but I'll stay on here as long as you want and ask questions. Thank you.
Yeah, certainly everyone's welcome to either continue putting stuff in the chat or unmute yourself and ask Kent directly. We had one other question that was in the chat that I just answered about whether you should put your data that you find your observations of butterflies on both eButterfly and iNaturalist. So we pull our data from both sources. If you put it in both places, like if you want credit for seeing a butterfly on both sites, when we pull that data, we're just going to remove the duplicate. So we're only going to use one site or one version of that data.
Yeah, I know a lot of people love to keep their life list on both sites, and it's totally, like you said, Dana, it's totally fine to put it on both sites. The thing that I just continue to tell people about eButterfly is that it collects what's like eBird does, it collects checklisting data. So we get your effort, like how many people were out there butterflying with you, how much time you spent, the distance you walked, and all the butterflies you counted. Like instead of just like, oh, there was a monarch here, it's like oh, there was a monarch here and there was seven of them. And so it's much more intense data. But if you also want to get credit on iNaturalist or you want to get help on iNaturalist IDing something, by all means, put it on both. It's totally fine. And we will deduplicate it and use the best available data So if there's the same monarch is only on as presence only on iNaturalist, but you counted it on the other one, we'd use the one that you counted. And if you have like also if you feel like it on both platforms, and I'm scared to say this, but I'm going to say it anyhow. You can use the at symbol, like at somebody, to get their attention. So if you have a butterfly that you're like, it's driving you nuts and you want a quicker second opinion because I might not get to it as fast. If you at me in my username, like the at symbol in my username, either on iNaturalist or eButterfly, I will get a message and you say at, KP McFarland. I can't remember what my username is, but it's something like that. At KP McFarland, can you help me with the ID on this thing? I think it's this, but it could be this. Like I will get that message and it usually will garner my attention because I want you to also be excited and learn too. And so if you have, don't try not to do it too much because I can get inundated. But if there's something really burning and you're trying to get my attention or somebody else's attention, go ahead and do that. Like it's great to have conversations on both iNaturalist and eButterfly about our sightings and our checklists and stuff. I mean, that's why there's the comment sections on these things.
So Kevin's got a question. Is there a brown color form for the caterpillars? He's run into both of those colors down in Somerset, brown and green.
They're both brown and green for all the species as far as I can tell. I didn't get into it today, but according to that paper, it has to be like up to maybe like I think it's maybe up to like second instar. You can tell by the amount of white on them because they still have that bird poop looking thing on them. It's how you can tell the species apart. But after that, no one knows how to tell them apart. And so if you find bigger caterpillars that are either brown or green, and I don't know why they're brown or green, to be honest with you. Maybe somebody does. You can't tell the species apart. And I actually have grabbed them and raised them on. So if I find them on like, oh, it's on a cherry tree, I'll raise them in a container on cherry in natural conditions and um, let them pupate. And then I keep them in my garage in the container so they're in natural conditions all winter long and let them eclose naturally in the summertime, in the spring or summer. And everyone I've done this to ecloses in May and is Canadian tiger swallowtail. I've yet to find a caterpillar for midsummer tiger swallowtail, but the only way I'd know is if I raise it and have it come out. And the midsummer tiger swallowtail has a delayed eclosure for some reason, and that's probably how it got to be. It's probably how it got to be isolated from Canadian tiger swallowtail is that it was maybe like Appalachian tiger swallowtail, 400,000 years ago, had a hybridizing event that caused it to eclose later and become isolated. Long story short, to say like you can't tell and you have to raise them to be able to tell, as far as I can tell so far, which is okay to do under natural conditions and can tell you a lot about what's going on and what food plant it is then. And incidentally, I go back then and I put it in iNaturalist as an adult as another one, and I go back to the caterpillar and I put notes in there and I put what the species is and why I know that. So then we have a record for the species and what it was actually feeding on. It just takes a lot of work, that's all. Great.
All right.
So any tigers you're seeing right now are definitely Canadian tiger swallowtail. There's an off chance that you could run into an eastern tiger swallowtail that came up from the south, but you're pretty much safe in May for leaning heavily towards Canadian tiger swallowtail.
Kevin says his brown caterpillars have been on the road, unfortunately.
That's why they're brown. They're mimicking the road.
I guess so. Camouflage for dirt roads.
Yeah. I found it, yeah, you will mostly find these a lot when they're wandering as like the last instar. They start to wander off their host plant and find a place to pupate, and I find them like crawling all over the place on my deck or like across the road, like Kevin's finding, and they're not on a host plant necessarily. They're not feeding, they're wandering for a place to pupate. And then it's still fun to let them pupate and then see who they are the next year, but you don't have the record for like what the host plant was because you don't know what they're feeding on.
All right.
Well,
Well,
hopefully everyone enjoyed the tiger tails. It's not easy, like it really isn't. But with a little practice, it you can ID a lot of them. And just like I said, just don't get worried if there's some you can't ID, like join the club. There's some we just can't ID. We're like, yep, it's one of the tiger swallowtails. I don't know,
mhm.
and it's okay.
All right. So, looks like we're probably reaching the end of our time. I don't have a lot of questions left. So, like I said earlier, this is the last webinar that we're doing for this year. Thank you. Many of you have been with us for the entire time, and if not, welcome aboard, even for the last one. You can reach out to me if you would like to see the other ones as well. I can send you recordings. So, like I said at the beginning, we'll probably be running this Lunch and Learn talk series again next year. I'm going to drop a form in the chat that you're welcome to add new topics that you're interested in learning about next year. I'll also send this in that follow-up email. And while this is our last webinar, our summer in-person events have finally been scheduled. So if you visit val.vtecostudies.org/events, you can find all of the events that are going on near you and across the state. The main ones that we'll be doing are called butterfly blitzes. This is going to be us going out to priority blocks across the state and doing like a fun little short surveys period where VCE staff will be leading teams of participants to go survey as many butterflies as we can. It's kind of like a little fun competitive event. But if you are, Terry level expert, or if you're just learning butterflies, have fun, come out, join us, and meet other butterfliers as well. So I'll drop that link in the chat as well. And we're kicking off our in-person season with a regular training, which is like an indoor and outdoor combined training coming up at Dead Creek Wildlife Management Area with VT coverts on June 6th.
Ooh,
So
that sounds
that is
fun.
coming up. I will also send those out in the follow-up email as well. And Kent, any last comments?
Go watch butterflies.
Perfect. All
Thanks,
right.
everyone.
Thank you all for coming. We'll hopefully see you this summer for one of those in-person events. And as Kent said, happy butterflying.
See ya.